David Klatzmann, also a well-known immunologist, clinician with long-lasting focus on T cells in different contexts, from infections to cancer to autoimmunity. And so he's going to now focus on his new or his target in the lab, which is to better understand specificity of T cells. So, David.
So thank you, Pramita. Yes, I'll talk now on the specificity of the T cell immune response. And Antonio already, mentioned some part of that specificity, that is the tight one, and I will mostly focus on what has been my interest for some time now, which is the fuzzy part of that specificity of the T cell response. So on adaptive immunity, what we learn and teach is that specificity and memory are consubstantial to this adaptive immunity. And this is summarized by the fact that, we all think that if we are vaccinated against measles, we should not be protected from influenza. And because memory largely depends on specificity, it means that overall specificity is the main property of adaptive immune response. So it's worth, spending some time trying to understand that. And when you try, do that, you can study it, at multiple levels. But the two I'm going to focus on right now is the outcome of the immune response, how we can analyze PD through the outcome, and also the specificity of the TCRs that are supposed to mediate this specificity. So first, on the specificity of the adaptive immune response, the outcome. So one of the questions you have to ask yourself if you think about adaptive immunity is, how does an immune system that is endowed with specificity and memory can handle, countless pathogens, okay? What we learn and teach is that we have a very diverse TCR repertoires. And in theory, you can generate, 10 to the 19, some have estimated 10 to the 61 different, TCRs. But we have only mostly, around in the 10th of the 11 T cells. But so that's obviously the largest possible size of an individual repertoire. But that's already a lot. 4 times 10 to 11 is roughly the same number in dollars that the wealth of Elon Musk, for example. So it's a lot. And we also, learn and teach that while you have all these different specificity, when you have an immune response against the virus, you have to have clonal expansion of the cells that are specific for these viruses for getting enough fighters to control the virus and later produce the memory cells. And indeed, to support this, vaccination with measles-derived antigen protects against measles. And altogether, there's a large literature to actually support this. But however, there are actually numerous inconsistencies that you can find, in the literature, in actually very good journals, which are largely overlooked by immunologists who are studying specificity of the T cell response. And I'm going to list a few of these, which I think are extremely important. So, for example, there's really a paper that I like to present a lot. It actually reports the story of a patient that had his entire T cell repertoire, TCR repertoire, coming from a single T cell precursor. This was a person with a SCID disease, and he had a reversed mutation of one of these T cell precursor. And in this paper, the authors estimated that from this single precursor, the patient generated about 1,000 T cell clones with unique T cell receptor beta sequences. This was, done at a time where it was not so easy to do real clean sequencing, but that's what they estimate. But that's really interesting, is that in fact this patient, so patient with SCID, usually, they have a terrible, outcome very early in life. They have tons of opportunistic infection. And if they don't get a bone marrow transplantation or now gene therapy, they actually die rapidly. But this patient actually had almost a normal life until, his 6th year. He generally, had no opportunistic infection. In fact, at some point, he developed a classical infection of children, varicella, which in the case of immunodeficiencies, it can lead to a very severe form of a disease, but he went very well, and he actually cleared the infection. In this patient, so all this is not actually due to a good B cell response, because in fact, in this patient, the B cell functions were low, and he had to actually receive IVIGs. And in fact, he was well for quite long. So again, this shows that with a very limited TCR repertoire, you can handle a large array of viruses this patient was exposed to. There are in the literature a lot of papers also that can make you think about this issue of specificity. For example, I had to quote this paper showing that there are a lot of so-called virus-specific T cells which have a memory phenotype in unexposed individuals. And that actually was published in a very good journal. And in fact, it's not just rare cells, but there was an abundance of memory phenotype cells, 20 to 90%, averaging over 50% in the CD4 T cells specific to viral antigen in adults who had never been infected. So how do you get, memory cells without, seeing the antigen you're specific for? These are questions I'll come back to some explanation later on. Then there is also this whole field of heterologous immunity that I discovered, I must say, quite late in my immunology career or life. I've been to tons of immunology meetings. I never actually went to one that had a specific session on heterologous immunity. Well, it's, to me, a very important field. It has been led for quite some time by Celine and Welch from the late 50s and until nowadays. They're still publishing on that. Their pioneering studies demonstrated that mice that were infected with one virus, for example, LCMV, they actually had altered or enhanced immune response to unrelated viruses, for example, Vaccinia or Pinchili or VSV, okay? And what they had, they were either having a faster viral clearance or an altered pathology, and they summarized this in one of their reviews, where they, for example, show these 5 different viruses and how infection, for example, with influenza actually improve the outcome of Vaccinia virus infection, and sometimes it goes reverse, it's altering this. There are also a lot of data on heterologous immunity in humans. For example, these are mostly epidemiological studies, but they are important. Studies conducted in children in developing countries show that there is a benefit in all-cause mortality and in infant mortality after measles vaccination, which was far over and above that which could be explained by preventing measles-related mortality alone. And there are actually dozens of papers on that. So if you're vaccinated against measles and you're in the developing countries, you're infected by many other pathogens, which sometimes can cause severe disease and death in these countries, you do much better if you've been vaccinated against measles. And this is not only, seen in developed countries. Actually, there's this beautiful study performed in Denmark, where they followed, 500,000 children that had actually contributing to over 50,000 hospital admissions for different type of infection. And basically, following this patient, they concluded that in a cohort of Danish children, receipt of a live MMR versus inactivated DTaP HIV vaccine was associated with a lower rate of hospital admission for any type of infection. So you can have empirical evidences for heterologous immunity in humans also. Now, you can also, in a functional way, and also going in the direction of what was showed by Celine and Welch. There are experiments, in humans that are demonstrating this heterologous immunity. For example, having had a cytomegalovirus infection in human enhances the immune response to influenza. This is a Mark Davis paper, a Mark Davis group paper, and it's a clear demonstration of that. And there are really no explanation for that is provided in this paper. And finally, again, still being a bit mechanistic, Celine and Welch and others, and I'll come back to this also later, showed that there is a functional part around this. And for example, here, they generated T cell line that were specifically activated and culture against the M1, antigen. And when after some time they come back and restimulate these cells with M1, they see that they can activate a large fraction of these. But if they come back and vaccinate again a totally unrelated, peptide from EBV, actually, which is BMLFF1, they can actually see a very nice small population of cells that actually does produce interferon that is binding the tetramer of that peptide. So these were, paper I wanted to highlight to just, challenge that there is only very specific responses ongoing in the T cell adaptive immune response. Now let's go and see what we can learn from studying the TCR, looking at this. So this is a typical scheme of a TCR showing, how the antigen is presented by HLA and recognized by the TCR with these two chains. And this kind of very classical representation, highlights a lock and key model that implicitly suggests a tight specificity. But we know, and in fact, sometimes we just ignore that, but we know that these TCR are not always so specific, and there's a whole literature on cross-reactivity. And what is cross-reactivity? It's the fact that TCR peptide MHC interactions are about conformation, and there is room for molecular mimicry. And this has been studied broadly by in a famous paper by Don Mason in Immunology Today, like more than 25 years ago now. Where going through multiple calculation, he came with the conclusion that T cells must be widely cross-reactive. If not, you cannot explain your results. So that one cells, he estimated that one cells react productively. It's not that just like bind a little bit of a peptide or things like this. It just react productively with approximately 10 to the 6 different MHC-associated minimal peptide epitopes. So to Don Mason, 10 to the 6 different peptides. There's another, important paper that has actually tackled more or less the same thing, but differently. It's a paper by the Udall group in 2009, where they estimated that, in fact, a single TCR recognize if you have a pool of random peptide will recognize 1 in 30,000 peptide. And in a universe of a much larger number of peptides, it means that actually T cells can recognize many different peptides. So what I wanted to highlight here is that when we speak about TCR specificity, we define it by the knowledge we have an interaction. We've made experiment to generate cells, clones, sequence the TCR, and we're just talking about what we're seeing, what we know. But we do this in the context of an ignorance of the other interaction that this TCR may have, as highlighted in this paper. So basically, what we do is when we're looking for our keys under the lamppost, because this is where light is. Okay, we don't like to study things we don't know, in fact. So now cross-reactivity, it needs to be defined also a bit better, because I would like to introduce a difference between cross-reactivity and polyreactivity. So cross-reactivity, for example, has been beautifully studied in this paper by Nelson and et al., where they've studied the response to the MOG peptide, which sequence is here. And they actually did some random mutation in the peptide to see, which mutation will conserve, the recognition by the TCR. And they came out with a result which showed that only 5 positions were really required for being recognized by the TCR. So what they did, they searched in databases if they could find, peptide that had this same, amino acid at this position, and they could find 3 actually that were derived from bacteria. And what they showed is that when they were, making, tetramers with these peptide, they were recognized, by the same cell and at the same frequency than the original MOG peptide. And not only they were recognized, but there was a functional relevance to this recognition because they could induce EAE in these in mice with this peptide almost as well than with the original peptide. So this is basically cross-reactivity, where you generate in this tight, peptide MHC TCR interaction something that more or less the same shape and is recognized more or less the same way than another unrelated peptide. But there are also, things that are a bit different and that are called polyreactivity, and we came to this, from the different, initial paper that I reviewed earlier, where we finally asked ourselves, could the same T cell recognize and be activated by antigen from unrelated viruses? We know a bit, Celine and Welsh did this experiment, but we wanted to investigate this in more details. And basically, most of what I'm going to show you from our experiment is in this paper that I'm mentioning here. So first thing is, we looked in database, okay? And we looked in the widely used database that we first curated to retain only the TCRs for which the viral specificity were defined by tetramer binding, okay? Which is a standard in immunology experiment. And we're very surprised to find that there are in databases a lot of TCRs which have been described by Lab A in Europe as being specific for CMV, but the same, I should say, this CDR3, but that has been described as specific for influenza in Lab B in the States, for example. And these were not just rare cells, they were frequent cells, and sometimes they were even, they had been described as being specific or binding tetramers for 3 different peptides. And then we turned to our own, data set that we were studying at the time. And indeed, we could find, among 13,000 different virus-associated beta CDR3s, that a lot of them were actually reacting to an antigen from a different virus. So, but of course, this was, looking at CDR3s and only the beta chain, which, of course, doesn't make all of the specificity. So we then turned to analyze a data set from a single cell TCR sequencing data set. And because of time, I see the time is running now. Basically, what we showed is that at the single cell levels, there were cells that were defined as binding, dextramers from 2 different peptides, for example. And we went up to the cloning, actually, of these TCRs to show functionally that they were indeed responding to totally unrelated peptide from different, viruses. And this is also in the paper. And these polyspecific or polyreactive peptide, because they react, so we can call them polyreactive. They're not all the TCRs, because some TCRs are also, very specific and cannot be activated with whatever we've tested at this stage. Now, also functionally, we've made this experiment, which more or less reproduces in a different way what Celine and Welsh did years ago, where we took CD8 cells that we activated that we selected after binding a dextramer for either a CMV or an EBV peptide. We cell-sorted these cells, and then we activated them either with CMV, EBV, or OVA. And basically what you can see here is that cells, although they've been selected for binding a given dextramer, they actually respond to both of them, but not to OVA. Now, again, to discuss the relevance of this polyspecificity, we showed, and for lack of time, I don't go into the details of this, but if you follow the dynamic of a TCR repertoire after vaccination against yellow fever, it's a public data set. You see that a lot of the cells respond not only to actually yellow fever, but also to many different antigen from many different viruses. And this is also true in our own data set where there were patients vaccinated against EBV. Also, during COVID, we analyzed the bronchoalveolar lavages from severe COVID patients. And when you look at the TCR repertoire of these cells, you can see that there are some large expansion that you can actually detect in these lavage. And when you look at what they are made of, they're mostly made actually of TCRs that have been assigned to recognize different viruses, okay, more than one viruses, so polyreactive TCRs. And finally, we're not the only one to describe these cells. For example, we know that in tumor-infiltrating T cells, there are a lot of cells that have been described as recognizing, viral peptide, although this is not a formal demonstration for polyreactivity. But also in this paper, again, for the Mark Davis lab, not going into the detail of this, it's a beautiful study, a lot of the single cell sequencing. They could finally identify a new tumor antigen and see that the cells were responding to this antigen, but they were equally responding to actually peptide from EBV and E. coli. Okay. So what can I conclude with these various observations that I've drawn on the screen? First, I'm not challenging that there are specific immune responses. If not, we would not be here to discuss probably, and we will not be protected by vaccines. But it doesn't mean that the T cell immune response in general are very specific or only specific. If we start looking away from the lamppost, we have to recognize the speed of TCRs and of T cell immune response should be somehow redefined. This specificity is not binary, okay? It's not there is one TCR that recognizes one peptide, but it's really probabilistic. There is a whole array of molecules that can be recognized. And in the end, whether you will be activated and expand will be very probabilistic. And this is the property of a system that is, structurally flexible and degenerate in its recognition. I also showed you that there exists a large population of alpha beta CD8 T cells that carry polyreactive TCRs that can respond to multiple viral antigens, but not all. And again, out of 160,000 unique sequence of that kind, we analyzed 24,000 actually could bind more than one viral derived peptide. So this to us defines a kind of a fuzzy specificity. So what's the possible role of these polyreactive TCRs in maintaining immune coverage? I want to go back to what I talked about earlier. How do we win our first battle against the virus when we first see it? Each time we're infected for the first time by a given virus, there is a kind of a race between the virus and the immune system because the immune system has to kill the virus faster than it can actually divide. But at the start of the race, the T cells that have a high, specific TCRs are pretty rare, and we know that we have to expand them. So it's actually quite a handicap if you think about it, because for the initial, period of virus expansion, you really cannot control it. And I think that these polyreactive TCRs with fuzzy recognition could provide a first line of rapid response to pathogen before a specific response could become efficient, a kind of somehow innate response within the T cell specific response. But there are important questions that remains around fuzziness. Phenotype specificity is probabilistic rather than binary. But how specific is specific enough? And I don't have time to discuss this. How can we reconcile fuzziness and self-tolerance? I do believe that, not shown in what I've shown you. Of course, these fuzzy TCRs can also recognize self-antigen. And you all know also that viruses are thought to be the trigger of autoimmunity. So I think it's possibly an explanation for that. And then I want to really ask a question, and possibly to Antonio, if specificity of a T cell response was so important, why are T cells not going through somatic hypermutation? Okay. Also, because we've talked about BCRs. There are also polyreactive BCRs that have been described, which more or less have the same type of property that I've defined for TCRs, and these have been reviewed here. And I'd like to conclude with this idea that maybe this fuzzy recognition is defining a kind of a pareidolia immune system. Pareidolia is a tendency to interpret vague stimulus as something known to the observer, such as seeing shapes in clouds, seeing face in inanimate objects or abstract patterns, and hearing hidden message in music. This is typically, seeing here a person. I don't remember on which planet this image has been taken, but some people have decided that this actually proves that there is a life, outside of Earth, planet Earth. And maybe, this is also a bit like how the immune system works. I want to stress that pareidolia was at one time considered as a symptom of human psychosis, but now it is seen as a normal human tendency. So with this, I'd like to just close and thanks, people from the lab, my former lab, now actually headed by Encarnita. Valentin did a lot of the work I presented, but all these people have been involved, and we've been collaborating with many TCR repertoire specialists. Thank you very much.
Thank you, David. I think we have questions already. Simon?
You had measles as an example. The effect of the measles vaccine may be just that a measles vaccine protects against the measles virus, and the measles virus infects B cells and depletes existing memory. So there was a paper in Science by Mina et al. That shows that the measles vaccine depletes existing vaccination status against lots of other diseases. So the measles vaccine may protect against this effect, and excess survival rate may come from this effect instead of T cell dependent effect.
Sorry, yeah, there's a
Yes.
Yes. I took measles because there were all these papers on the measles vaccination defining heterologous immunity. But yes, well, it's a possibility, but again, it's seen. There are a lot of experimental data with other viruses, in mice where that cannot be explained, just by this kind of infection of B cells.
Other questions or comments? Jamie. Jamie Scott. And then Jamie.
Thank you. Thank you for this wonderful talk. Yeah, so I just wanted to ask you a little bit more about what you mentioned earlier in your presentation, where you showed that a few residues in a peptide sequence were what I would call, from the antibody peptide world, critical binding residues, where they are responsible for affinity. And so I just like to say that and ask you how you think of this. So when you're talking about these, quote, polyreactive clones, are you just basically saying that they have shared critical binding residues, so a shared mechanism of binding, and in the background of different, other non-binding or non-important residues, as opposed to what I consider polyspecificity, where if you did a replacement series on the peptides, you would not necessarily see critical binding residues because there's a combination of residues that have to be together to create the effect. And especially with T cell receptors, as opposed to antibodies, where you've got, 2 or 3 logs difference. So you see huge differences in binding. So it's pretty easy to clearly define a critical binding residue and therefore shared mechanism of binding. Here with TCR, because you have multiple T cell receptors binding to multiple MHC molecules, et cetera, not so high a threshold for a reactive response. So I'm kind of wondering, in those two worlds of what I would define as specificity, but a degenerate background, and polyspecificity, where you really don't see critical binding residues popping up so much. Where do you fall in those areas?
Yeah, well, thank you for a question. Yes, the cross-reactivity, the paper I described, is really, again, showing that there are critical residues, and that basically the shape of the peptide that interacts is kind of fixed. What is in between, except of course if you have very hydrophobic, hydrophilic peptide could change it, but it's not so important. So you then find very different sequences, all together from bacteria or whatever other agent that have the same 5 critical, peptide, and so they bind the same way. So it's really the same binding, and in fact, it goes all the way to being able to activate the T cells the same way and generate the disease that is supposed to be triggered. I think polyreactivity is something very different. I think these are TCRs that somehow, for a structural reason, can accommodate a lot of different things, in a pretty loose manner, probably with a lower avidity. These are speculation, but these TCRs, we studied in the paper by Valentin. We showed that in fact they are positively selected in the thymus. So not only they go through selection, but they are enriched actually after second. We also showed that altogether these TCRs have a higher probability of generation than the overall population of TCRs that are not defined as polyspecific, which somehow indicates that they provide some kind of advantage if they have been kind of like selected. So we don't know exactly. I cannot answer your question precisely because we've not done these studies. What we've just showed is that we've cloned some of these receptors, the two chain, express these TCRs in reporter cell lines, and show that not only they bind tetramers with different peptides totally unrelated, but they respond. They are activated. So that's all I can say. And the whole point of this talk is just to say, okay, fine. It's good to study specificity and how we can make better vaccines with better responses at the T cells, at the B cell response. But and especially we come from a background that we're studying autoimmune disease, and we are of course very interested in the fact that these polyreactive TCRs may also at some point play some role in triggering autoimmune disease. But that's speculation.
Thank you.
Jamie, either?
I actually have a very closely related follow-up question. So I've also seen these, high P-gen TCR betas popping up in a lot of different reactivities, and I've always wondered, what's the balance between, is this, as you say, like a promiscuous beta chain that can bind to lots of stuff, or are these beta chains that get made a lot and occasionally will coincide with alpha chains, which happen to be specific to different things? Obviously, when you've got a whole clone, but can you speak to broadly how much is which?
Yeah, but they're selected, these T cells. Okay, so at some point they have to bind to something in the thymus. If they're not binding to something, they will not survive and will not be selected. So they recognize some things. They go through the selection process, and then when you find them in the periphery, you realize that you can't find just one very specific thing to which you respond, but you see a vast array of things to which you respond. And by the way, I want also to maybe specify here that Yuval and co-authors said that you can respond to 1 in 30,000 different peptide if you pick up randomly. But what we looked at is really 12 different antigens. So it was very unlikely that we could find TCR that responds to 3 different of these just 12 peptides that we were testing. So I can only speculate here, and I think they're selected. That's for sure. They actually are positively selected, and they even are enriched from pre-selection to past selection based on CD3 and double positivity and then single positivity. So they recognize something, and in the periphery they are actually recognizing more because they've been actually described by various scientists in their experiment. They pop up in their experiments, and they actually put them in database as specific for CMV or specific for EBV because they've done experiment with EBV or CMV, and this is how they assigned the specificity.
More questions or comments? So when you talk about this link between polyreactivity and autoimmunity, so how do you see this as a trigger, as in fact inflammatory process that maybe expand the antigen specificity or the inflammation?
Yeah, so the activation of a T cells requires the stimulation of a TCR, but also integrates the stimulation by a lot of different other stimuli, like cytokines, co-receptors, et cetera, et cetera. So I think that every one of us has an history of an antigenic scene, as we say it, where we've responded to some viruses. By chance, maybe we've expanded some clone of polyreactive T cells. We know that once a T cell has been activated, it's usually easier to be reactivated. And I think that if it happens that these TCR by chance also recognize with some degrees of avidity some self-antigens, they may at some point trigger some autoimmunity. But we're trying to study this and show this. It's just speculation at this stage.
Because you made the link between the TCR polyspecificity, the BCR polyreactivity also that is well known. So we know that the B cells and T cells are not selected the same way. We all believe, or at least I believe, that T cells are much more strongly selected in the thymus. B cells goes through different steps over time in different tissues. And so again, here polyreactivity and the role of these natural antibodies that were associated to polyreactive BCRs, how you see this with the T cells and the TCRs?
Again, as I said, if we see that these TCRs have high PGen, for example, higher than the overall population of TCRs, it implies some kind of selection. We know that our immune system has been, is somehow mostly selected by our interaction with viruses because we have to survive these infections. So I like the idea that because we see, and we define the polyreactivity based on viral antigen because there are many more TCR specific to viral antigens in the database and they are antigen specific for something else. Okay, so this is why we see them popping up so easily. But I guess this is what has actually somehow probably selected these TCRs if they have this higher PGen. And so I think that they must participate to the overall immune response to viruses. And of course, we have an innate immunity that protects us at the very early stage when we interact with some pathogens. And then we have these very specific immune response that are very protective and even are those that mediates protection after vaccination. But in between, when you're infected by influenza, I mean, you have just a few days to react before you're dead. This is why older people or really young children die of influenza. And if you don't die, it's because you have actually mounted a very good immune response that can eradicate the virus. But in these very early days, it's critical. There is a race, okay? And if you just have to find and expand and select these rare cells that are very specific, I believe you could be in bad shape. And if you're helped by a kind of some semi-innate T cell response by TCR that are not as selective, have a lower avidity, but still can react, you may actually contain the infection until you have enough of the very specific fighters.
Great. Thank you. Thank you, David. Thank you, Antonio, and thank you, the audience, for the questions. So now we have a small break, 10 minutes break. We can reconvene in 10 minutes. So I'm closing the session 1, sorry, and then Justin will open the